join tonight. I think you're going to really enjoy this hour session because Dr. Schug is working on something that I think is very near and dear, certainly to my heart and to most of us. He's going to talk about the role of diet and obesity and alcohol on breast cancer growth and spread. And as I sit here drinking my glass of wine, I am going to be very interested in what he has to say and probably feel maybe a little bit guilty after he's done. But I do think it's going to be an interesting talk. I have the pleasure of being the advocate in science, the Komen advocate in science that worked with Zach on this application. And it's one of the great opportunities of being a Komen advocate in science. In fact, as I look through some of the participants, I see several of my colleagues. I see Roberta Albany. Hi, Roberta, and I see Jamal Rivers, and there may be a couple of others that I have not caught. But Komen Advocates in Science is an organization within Komen that brings the voice of the patient to research. Komen requires that a patient advocate be involved in reviewing grant research proposals and have a say in deciding how those research dollars are spent. We also require that patient advocates serve as a vital part of a researcher's project team. And it's one of the things that I think kind of separates Komen from some of the other granting organizations in that they really see the value of bringing the patient voice into research and making certain that that research is ultimately translatable to the clinic and ultimately helps patients. If any of you are interested in learning more about Komen Advocate in Science, it's really easy to find out more. You just go to komen.org. You click on the Get Involved tab at the top of the page and select Become an Advocate, and you can learn more about advocacy opportunities at Komen. During this hour, we've got a chat box. I already see that somebody has actually plugged in, and Elizabeth's already put in the link for the run sign up in the chat box. We'll be monitoring that, so please, as we go through this hour, type in your comments, type in your questions. I may interrupt Zach during his presentation, so we can get some of these things answered. If there are questions we can't get covered in this hour, we will definitely follow up with you after the session. So as I said, Zach's work involves a better understanding of the role of obesity, diet, alcohol, and some of the metabolic things that go on in a tumor that helps that tumor grow. And Zach, I thought that maybe before you start telling us about your research, you might want to maybe share with us what was it that made you decide to become a researcher in the first place, and why did you decide to specialize in breast cancer?
So first of all, thank you so much for this opportunity, and it's great to see so many people here and ready to learn a bit about diet and cancer and all these fun things that I've been working on and trying to figure out. So yeah, I got into research because I've always had a passion for sort of learning about nature and biology. I grew up out in New Jersey in the Pine Barrens, and my backyard was a giant forest that I sort of played in my entire upbringing. So I was very close to sort of nature and to biology itself, and I was always sort of fascinated by it. And then when I went to school, I took biology as a major and actually went to St. Joseph's, which is a big botany school. And I did a lot of plant biology initially. It had nothing to do with cancer research. And it wasn't until I went to Scotland that I got really interested in cancer research and fascinated by sort of the diversity of it and the challenge of it. I always am looking for new challenges as well. And the diversity as well. So just like the nature is very diverse and all of its different wonders, cancers are extremely diverse as well. And I had some family members that have died from cancer. So both my grandparents on my mom's side died of cancer, one of lung, one of liver, and then my dad's mother also died of lung cancer. They were heavy smokers, most of them, so likely that was the cause for them. But it was a very real thing for me because it all happened when I was about 10 or 12 years old. And so that sort of impacted my life at that age when you're sort of just starting to understand life and sort of mortality. It became really impactful for me. And it was something I sort of took upon and said, you know what, I am going to go into cancer research. And that's what I took on when I went to Scotland. And then in Scotland, what got me into breast cancer research was, again, that diversity, that sort of really interesting that there's so many different types of breast cancers and so many different subtypes and all these other things going on with it. I thought that was really fascinating. And I was also always really interested in metabolism. And And with breast cancer being in a sort of fatty tissue of the breast, the lipids and the fats there and the interactions with the tumors themselves became a really interesting sort of thing that I wanted to learn more about and try to understand, and then hopefully develop the new therapies for, which we're doing now. So, so that's sort of how I went from like being just interested little boy playing in the forest to sort of a cancer researcher here at Wistar. It's just been a long life passion of learning about life and biology, really. Life sciences have always interested me.
Oh, terrific. Thank you for sharing that. Why don't you take over the screen now if you want to put up your slides and tell us more about what you're working on.
So it just needs to be enabled, Elizabeth. There we go.
And there we are.
Okay. And so can everyone see that pretty decently?
Perfectly.
Perfect. So that's great.
I'm hungry.
Yeah.
A little broccoli, some blueberries. Yeah.
Salmon.
So today we're really going to talk about how a tumor feeds itself. In particular, we're going to focus on breast tumors. And I have this idea that we can basically hit cancer's food supply where it hurts. So if we can figure out what a cancer cell is feeding on, and we can take it away, and hopefully we can use this as a new therapy. But at the same time, what are the things that we can do? You know, what are the foods that we can eat that could support what we're trying to do? Okay, so cancer is both a genetic disease and a metabolic disease. A lot of people think about it as this first part, the genetic part, because they know that there's mutations that happen in tumors. But they also don't realize that cancer was, for a very long time, considered a metabolic disease. And I would say from the 1920s until about the 1970s, cancer was considered a metabolic disease, and mostly because of the really drastic changes that occur in the metabolism in a tumor cell. And what I mean by metabolism is just what a cancer cell is taking up and using as its food. You know, because it needs energy, it needs building blocks to keep growing. And we'll talk about that more in a couple of slides. But, and what it really is saying is that there's really two parts to a tumor. There's the nature of the tumor that I call it, and there's the nurture of the tumor. And I think all of us have heard about nature versus nurture and bringing up children and these other things. But the nature really describes all the mutations in a cell that cause it to become cancerous. So this would be, in breast cancer, for instance, it would be a mutation in the HER2 gene that would cause HER2 breast cancers. Or it could be activation of some other type of signaling molecule like PI3 kinase, whatever. But these are the mutations that really are the driving force behind the formation of the tumor. But one of the major things that also affects tumor growth and allows it to grow is the nurturing of the tumors. And this has to do with the nutrients that are available to it. So things like proteins, fats, sugars, oxygen, vitamins. And all of these things, and this is a very important part of the talk, are delivered to the tumor through blood vessels. So these are your arteries and your veins here. And these are like the little nutrients. So picture that as a sugar, and that's a fat, and maybe that's a protein, something like that. And what happens is that your blood vessels deliver all these into the tumor, and the tumor cells feed on them, right? And they also feed on a very important thing called oxygen, which we're going to talk about today as well. So oxygen is also really important. We all know we need to keep breathing and keep taking in oxygen and stay alive that way. And cancer cells are no different. But what can happen is that tumor cells can develop a different appetite for different nutrients than, say, a normal cell in our body does. And what's nice about that for us as researchers and as people looking for therapies is that we can maybe exploit that difference as a potential metabolic vulnerability. So as I said, cancer is both a genetic and metabolic disease, and cancer cells really require two things in order to grow. So they need building blocks and they need energy, right? And the way I always describe this is that if you were trying to clone yourself, which many of us would probably want to do, but if you were to do that, you would need a lot of energy to do it because it would take a lot of effort, but you would also need all the materials to make another one of you. And cancer cells are no different. So as a cancer cell divides and proliferates into two daughter cells, it uses a lot of energy and a lot of building blocks. And the way the cancer cell gets them is through the diet, is through feeding itself, through taking up nutrients. And we all know that cancer is a disease where there's proliferation. And if we can stop cancer cells from dividing and growing and proliferating and doubling and doubling and doubling, then we can fight cancer. And so for me, it just seems very logical that we can go after the metabolism of a cancer cell to try to target it and keep it from growing. But one of the things that's really been interesting to me, and what my lab has been really focused on in the recent years, is that the nutrient availability to a cancer cell can actually be really different depending on where it is within a tumor. So if you think about a tumor as a single mass, right, of cells, the nutrients that are available in some portions can be very different from the nutrients that are available in other portions of the same tumor. And this is a really fascinating thing that I hope we can exploit for therapeutic purposes. And so here's sort of an example of that going on. So there's our blood vessel, right, that feeds the tumor, you see here. And you can see that there's branches coming off, like these would be capillaries going into the tumors. And then here we have our tumor mass. So we have a malignant solid tumor. And tumors have areas where they have normal oxygen, and then they have regions where they have very low oxygen. And the reason for this is just because the blood vessels don't quite make it all the way up to the top, right? And so what happens is that these ones here have lots of oxygen around. So there's a high oxygen level delivered by our blood. There's also a high amount of nutrients available to them. And then what happens is they feast on that here, and they take them up, and they're using them to grow. And then these ones here have a little bit less to feed on, right? Because they're a bit further away from the source of the food. So this one, you know, leaves whatever's left, and this one will start eating it, and so forth and so on until you get to this region back here. And what we call these is the hypoxic region, or the hypoxia region of the tumor. And what it really is it has low amounts of nutrients available, and it has low amounts of oxygen available to it. And so these cancer cells back here are really stressed out. They're really struggling to feed themselves. They're really struggling to get enough oxygen to survive. And so as you can see here, you have this sort of gradient of nutrient availability and oxygen availability across the tumor, depending on how vascularized it is, so how many blood vessels are feeding it, okay? And then here's a real life example of a section through a breast tumor. And this dark brown stuff is actually labeling the hypoxic regions. And so this dye stains these regions here that you see here. And so here you can see there's no stain, so that means there's lots of oxygen there. So that would be these regions. And then here we have low amounts of oxygen, which would be these regions. And then we also get regions like this, where the oxygen and nutrients are so low that these cells have died. So this is called necrotic regions, is the term that the pathologist will use. It calls it necrosis or necrotic regions. And so it can happen and become so severe that there's not enough nutrients, there's not enough oxygen, the cancer cell just dies, which sort of makes sense. Cells do need some level of nutrient availability to them. But what we were really worried about were these cells here, because they often can become malignant. They also lead to more aggressive cancers, because if you think about it, they're fighting for their survival back here. They're trying to survive any way they can, and they adapt and they evolve while they're back there, and they sort of change their ways so that they can live and become more sort of resistant to low levels of oxygen, low levels of nutrients. So just to recap, we know that these cells here have plenty of oxygen available to them, and they're like basically living in a supermarket. So they can feed on whatever they want because the blood vessel is dumping it right there for them. And those cancer cells have plenty of oxygen and plenty of nutrients such as sugar, and they can use them to make those building blocks I was talking about and to supply the energy to sort of create new cells and keep growing. But back here, we have these hypoxic regions where we don't have very much oxygen, so they don't have enough oxygen to use. And they don't have many nutrients like sugars and other things for energy and growth. So what do? Well, they need to find other nutrient sources. And so they're basically trying to survive in a place like this as opposed to a place like this. And you can understand how that can come with many challenges to the cancer cell. And what we know is that these cells, as I mentioned just previously, they're also the ones that tend to turn metastatic, which is really the focus of the Komen grant that I have, is to really go after metastatic tumors. Because these ones back here are sort of fighting for their survival, they learn to adapt and survive almost any means. And they also become malignant and metastatic because they want to get to this blood vessel so that they can get some nutrients. And so what they do is become very migratory and start trying to get to there. And this, as you know, the migration and metastasis, you get the cells moving towards the blood vessels, and then they leave the blood vessels and go to somewhere else in the body. And so what we really wanted to do was fight these cells and try to go after this population. Because many of the common chemotherapies and targeted therapies, they work really well in these cells because the drug gets in there easily, not much drug gets back here. We also know that many of these cells are highly proliferative, so they work really well with chemotherapies and radiation therapy. But these ones back here are often the ones that are resistant to radiation therapy, resistant to chemo, and they're the ones that are sort of sticking around, these persisters that are sort of the nightmare of cancer for all of us. So we're really trying to figure out what these tumor cells are using and how they're surviving in conditions like this, so that we can maybe fight them and kill them off as well. So what we found out, and the rest of the talk will be about this one sort of nutrient, which probably many of you haven't thought about, but you will probably have heard of it. So what my lab found out is that instead of using sugars and other things back like they do here, so all the regular nutrients that would typically be supplied, all of them are used up. But one nutrient that makes its way all the way to here is called acetate. And acetate is used as an alternative nutrient source to help these cancer cells survive. Now, acetate, many of you know it maybe as acetic acid, and it's what basically gives vinegar its taste and smell. So that's the 5% acidity, that's actually acetate in the vinegar. And the acetate isn't used by these cells. These cells don't touch acetate because they use all the other nutrients that they can get, like the proteins, the fats, the sugars. They just feast on what's ever available. And so they actually don't use the acetate. And so the amount of acetate that's here is the same amount that's here because none of these cells use it. It's only here that these cells are able to exploit it and use it as an alternative nutrient source. And so we thought this was fascinating. And it was something that no one had really known about. We found this out back in 2014, and we published a number of articles. And what was really neat at that time was that the same time our paper came out, there was 2 other labs down at UT Southwestern in Texas that also found that acetate was really important for tumor growth. And so it was really neat going to the conference and interacting with them, and all of us sort of bouncing ideas, really excited about this idea that, you know, these tumors are actually feeding on acetate and surviving on acetate, which none of us really thought about. So that became a real strong focus of my lab. Can we somehow block the ability of tumors to use acetate as a nutrient source? And can we really go after these sort of hypoxic cells that really are the aggressive cells within the tumor, and the ones that are creating resistance to therapy, and also metastatic disease? So basically this was our scenario. These ones back here are basically saying we love acetate, and these ones up here are saying we love sugar and fats and proteins. And so there's this real difference between what's going on here and here. And what happens now is that these cells use acetate for their building blocks. So you can turn acetate into fat, which we'll go over in the next few slides. And you can also burn acetate for energy. So it's basically everything a cancer cell needs. It can survive on that. And it can then drive its proliferation, or at least let it survive for a little while until a blood vessel is able to come along and reestablish its normalcy, the normal sort of amount of nutrients and oxygens available. So acetate's really seeing these cancers through these sort of troublesome periods when they're in the tumors. So now I know what you're all thinking. You're all thinking that acetate gives you cancer, but it doesn't, right? So I just want to lay this disclaimer here. Our research didn't really show that acetate gives you cancer. What it does suggest is that acetate may be something that a cancer cell could use when it's in trouble. And this is a really important concept because it gets back to this idea of cancer risk versus cancer progression. Acetate's not a cancer risk, so, you know, please keep having your salad with its balsamic vinaigrette. It's not going to hurt you. But you do want to think about things like this when it comes to treating a cancer. So in other words, when you have a cancer and it's progressing or growing, maybe it's using acetate as a fuel source. And this could very well occur because hypoxic regions happen in probably every single solid tumor. So regardless of the disease, whether it's breast or pancreatic or colorectal. So almost every solid cancer has hypoxic regions. But I just wanted to say, don't think that acetate's cancer causing. It's not. I just wanted to throw that out there. So we started to develop inhibitors to see if we can target the enzymes that let these cancer cells use acetate. So can we block these cells from using acetate by adding like a drug to the tumors? And what we found is that when we inhibit the enzymes that are able to allow the cancer cell to use the acetate, we can block tumor growth. And we also did this not only with inhibitors, but we also take the gene out of the cancer cell as well using CRISPR technology, which I won't get into, but it's a really neat modern technology that lets us take genes out of the cancer in a dish and then grow them and see what happens. And so if we either take the gene out or if we drug it using a compound, we're able to block tumor growth really nicely. And I'll show you some results. This is the only results I'll show you today from the lab, but I think it's very meaningful. And so here we're looking at tumor growth in a mouse model. And this is a triple positive tumor. So this is ER positive, PR positive, and HER2 positive. So triple positive breast cancer. And in the black line, you can see what happens when we don't treat, or we treat with just the vehicle. You can see the tumor grows and gets larger and larger and larger. And the arrow shows when we started the treatment with our inhibitor. So this is our inhibitor of the acetate metabolism, and it blocks the ability of the tumors to use acetate. As soon as we add it, boom, we stop the tumors from growing. Many tumors stopped growing altogether. Other ones actually regressed. And so here's some images from the tumors from the mice. You can see that this one shrank to a very tiny level. This one was sort of middle, and this was a middle, this was middle. This one actually fully regressed. So we actually completely regressed the tumor in this mouse using this acetate inhibitor, which was really fascinating to us. Because one thing we were thinking about was that it was just important for the hypoxic regions, but there may be some type of feed-forward mechanism, or there may be tumors that are actually addicted to acetate regardless of the blood vessels. So this is something we're really trying to pursue, because it was unexpected, but really, really nice for us that we were able to see complete regression. And we're really trying to understand exactly what's controlling that scenario. But you can see right now, when we add this drug to our mouse models, we're able to completely block growth, and in some cases cause tumors to completely regress and disappear. So this was really promising for us. And we're trying to develop more and more of these inhibitors right now in collaboration with other people at Wistar. And we're also trying to find big pharma and biotech companies that want us to go to collaborate with us and maybe move it into clinical trials in the next couple of years. So we're really hopeful for these inhibitors, and we'll see how it goes. So I guess a question that all of you are thinking about is, well, where does that acetate actually come from in the tumors? And this is actually a really interesting question. It was something I actually didn't know when we first started talking about acetate metabolism in tumors and what's going on. It was something I didn't know. And so I started looking into it. And it really comes down to this very important thing that we all hear about, is you are what you eat. And then we started looking into how breast cancer cells feed themselves. And we all know that things like too much sugar are bad for you. You definitely don't want to be eating big fatty steaks all the time, or eating chocolate bars like this little boy. We probably want to be somewhere down here where we're having a bit more of a healthy diet, but as long as we have a varied diet, I think it's going to be okay. So where does that acetate come from? So acetate is almost exclusively produced by the bacteria in our digestive tract. So here we can see our digestive tract, and this is sort of your intestines here. And inside these intestines live a whole bunch of bacteria called your microbiota. And you may hear about, taking yogurts for the good bacteria in your gut, and all of those different things that are really popular now, and they are really good. And what happens is that the bacteria here in your gut are the ones that produce acetate in our body. And there's really two foods that produce lots of acetate in our bodies, and we'll go through both of them in the next few slides. The first is sugar. And we know a lot about, from the press and a lot of the news outlets and all these other things, that there's a strong link emerging between the consumption of sugar by tumors and the use of cancer cells of using sugar to fuel their growth. But in particular, we were really interested in one type of sugar, and many of you may even know it. So there's a particular type of sugar called high fructose corn syrup, and it's present in a lot of different foods. The most famous of which is this guy right here. Everyone knows a Coke can. I actually had a Coke tonight with my pizza, so it's okay to have it, I think, in a little bit, but overdoing it, I think, is too much. And then here you can see right on the label, high fructose corn syrup is an ingredient. It's the second highest ingredient within the soda. And high fructose corn syrup is actually found in a lot of different products that you can see here. And this is a very small list, but it's something that a lot of manufacturers of food will use to make it a bit more palatable. So it's a bit sweeter to the taste. And it's an inexpensive way to increase, to get that sugar kick to all of your different products, whether it's cookies or whipped creams or what have you. And what happens with your body when you digest high fructose corn syrup, oops, there we go, is that your body will turn that sugar into fat. But what we found out with collaboration with University of Pennsylvania people is that the way that fructose and sugar is broken down into fat and made into fat is not exactly how we always thought. So we thought that the liver would just take the sugar and turn it into fat. And that's sort of what everyone accepted. But in a recent study that I did with Katie Wellen, who's a researcher at University of Pennsylvania, and you see I was part of this study as well, which was published in Nature just a couple of months ago, what she and I found out is that when you consume lots of fructose, so when you drink lots of cans of Coke and you eat a lot of these foods on a daily basis, you get this large bolus of fructose that eventually gets turned into acetate, right? And all of this happens through those bacteria that live inside our tummies, called the microbiota. And so the microbiota will break down fructose into acetate. And then what happens is that that acetate gets delivered to the liver and made into fat. So here, I'll take you through this animation I made to sort of take you through how it happens. So here, picture yourself drinking some Coca-Cola. So that fructose makes it all the way down into your tummy, and those bacteria there will start taking that fructose, that high fructose corn syrup, and they will break it down. And so those bacteria will break down that fructose, and this leads to production of acetate. That acetate can then be taken up by your livers, and that's turned into fat and redistributed into the body, and then you get weight gain and obesity even. But obviously, one of the other things that we were really interested in is what the cancers were using with the acetate. So if we have a lot of fructose and a really poor diet, we may be fueling acetate production, which is then feeding the tumors in that way. And so you can see how things like controlling your diet can have a real impact on what's happening within the tumor. So I said there were two sources of acetate. So the first one was the high fructose corn syrup. The other one, as Meryl sort of alluded to at the beginning of the talk, is alcohol. So alcohol and breast cancer have this sort of storied history of being a high risk factor for a lot of different things, particularly breast cancer. And I think some of the data is emerging that there's definitely seems to be a higher risk when you are a heavy drinker, and in terms of increasing your risk for developing breast cancer, among other cancers, particularly the upper air digestive tract. So you can imagine like your throat cancers and esophageal cancers, these other ones that are, where the alcohol is directly being exposed to the tissues. But so the way that alcohol is turned into acetate is by our livers. And the way I always describe this is we have a few drinks, we may be a bit drunk or tipsy or, have a little too much. And the way we go from being drunk to being sober is that our liver converts all of the alcohol into acetate. And that's what we do. And so a person that's drunk has high blood alcohol levels. That's why you blow a breathalyzer, and the cops, that's what they look for. But if you're sober, you'll have very high blood acetate levels because all that alcohol gets converted into acetate. And that's how we sort of sober up. But what happens is that the amount of acetate in your blood goes up 20 times its usual amount. So now it's really concentrated in the bloodstream. And as we talked about earlier, if there's a tumor that really uses acetate, you can see how the alcohol conversion to acetate could be really fueling its growth. And so it's something that we're sort of investigating now and trying to get to the bottom of is after a tumor is existing and there's alcohol, does it actually promote growth of the tumor through acetate? And so that's something we're trying to figure out. As I said, there's this long and storied history between alcohol consumption and breast cancer. And this was a study in The Lancet, which is a very prestigious journal published a couple of years ago. And what they did is what's called a meta-analysis. And a meta-analysis is sort of a big word. What it means is that it's an analysis of other analyses. And what I mean by that is each of these black circles that you see here, each of these dots, represents a different study that's been published in the last, let's say, 50 years or so. And its findings. And so what they said here is this is the number of units per day, so not drinks, but units of alcohol per day, and then the increase or decrease in relative risk for breast cancer. And you can see that the one would be no change in risk whatsoever. But as you can see, as there's increased consumption of alcohol per day, the increased risk for breast cancer goes up and up. So eventually, once you hit a certain level, it just sort of levels off. You don't really study too many, times where people are drinking more than 10 drinks a day. But there's this trend that suggests that there's this link between alcohol consumption and breast cancer. But what you can see is that some of these are below the line and some of these are above the line. And this really gets to this idea that it's really hard to say that alcohol causes breast cancer. All we can say is that the increased consumption correlates with breast cancer. And so this is a really important concept for a lot of people to know about, is that although these huge studies that look at these thousands and thousands of individuals, it's all correlation, and it's really hard to establish causality. In other words, is it the alcohol that's causing the tumor, or is there something else associated with it? And then a lot of that has to do with just, normalizing the data, and it's human data, and people report what they had or what they didn't have. And there's also the social stigma of alcohol consumption. So people may underreport what they're having because of the social stigmas associated with over drinking. So it's really hard to get a nice robust number for this. But I think by and large, you can see that there is a trend. More studies report that there is an increased relative risk. But the other thing I wanted to point out to everyone was sort of interpreting these relative risks things that you see here, where if the relative risk is 1.2, that means there's a 20% increase in the risk. But that doesn't really mean that you're 20% more likely to get cancer. What it means is that since breast cancer has about a 12% risk over a lifetime, it's a 20% increase of 12%, which actually is 14%. So whenever you see these relative risks, always take them with a pinch of salt and realize that they're talking about increases of something else. And so, so that's something I just wanted to point out to you, that although 2 drinks a day increases by about that 20%, it suggests that there's only an increase maybe of 12% to 14%. It's not huge, but there is an increase. But what was really interesting by the study is that they found that there was no safe level of alcohol consumption for any outcome. And they looked at a lot of different disease indications, breast cancer being just one of them. And the only time that they never saw alcohol being a problem was when there was 0 to 1 drinks per day. So what they're really suggesting is to really minimize the amount of alcohol you're consuming if you want to reduce your risk for all these different diseases and pathologies. So my lab, as I said, is pursuing the idea that very high amounts of acetate in the bloodstream that happen after you drink alcohol contributes to the risk between alcohol and breast cancer. And the idea that we have is that alcohol and high fructose corn syrup are broken down by your microbiome. There's bacteria that live there, and then converted by your liver into acetate. And that acetate is then fed into the cancer and helps fuel its growth, particularly those cells that are those aggressive ones that sit in the hypoxic regions. And so what we really don't want to do is not fuel those cancer cells and sort of take that away from them. And although alcohol may not be causal in nature, the data suggests that it may be, but it is a correlation at the time. But what we do want to find out in my lab is that once a cancer is formed, we believe that the alcohol consumption and high fructose corn syrup, like a poorer diet, such as a high fat diet, high sugar diet, leads to the production of acetate, which then fuels aggressive and malignant breast cancers to grow. And so we're really testing all of these things in the lab, and we're developing models for alcohol consumption and high fructose and high fat diet consumption. And we're really monitoring these acetate levels, and then we're testing our inhibitor at this stage, and then also using genetics to remove the acetate enzymes from the tumors and see if we can block it that way. So that's really what our research is doing. And more specifically, we're also really interested in metastasis. And this was the research that was funded by Susan G. Komen. We're really interested in brain metastases. And the reason for this is it's quite interesting that the brain is the only organ that gives off acetate as a waste product. So every other organ in your body, your skeletal muscles, your heart, your liver, your colon, every other organ, your lungs, they all actually take up acetate from the blood. And what we found very recently is that the brain produces it as a waste product from the breakdown of other things. Now, I know that I told you earlier that the bacteria in your gut are the ones that produce the acetate in our bodies. And by and large, that's really true. The vast majority of the acetate, I would say 95% of it that's made, comes from the bacteria in our guts. But there is a little bit of acetate that's produced by the brain. And now you can imagine that being a sort of bad scenario, right? If you have a tumor that was really like to use acetate and figured out how to use acetate by living in those hypoxic regions, it then became aggressive and metastatic and left the primary breast tumor, entered the bloodstream, and then sort of settled into the brain. Well, now the brain is actually producing acetate, so it can feed the tumor. And this is a nightmare scenario, right? So the brain is actually producing exactly what the cancer cell needs. And so this is one thing we're trying to test, because right now we think that when a cancer cell enters the brain, it feeds on the acetate that's there. In other words, what one cell's waste is another cell's treasure, if you want to think about it that way. So the cancer cells are exploiting the waste product of the brain to its own means so that it can fuel its growth and its energy. And can we take our inhibitors and block the growth of these brain tumors? Or can we even block the ability of a cell to metastasize and settle in the brain if we block the ability of it to use acetate? So that's really the couple of things that the Susan G. Komen project that I'm working on is really focused on, because it has a strong lean towards understanding metastasis, but particularly from the point of, can we stop its use of acetate as a means of fighting it? So in the last couple slides, I'm just going to introduce diet and cancer, because I think this is a really interesting topic, particularly for the patients. And I think this is also where patient advocates, I think, can be a strong part of my project. Because it's not something I can sort of test, and it's not something that we can really look at, because it's really hard to control. And so what do we know? So we know that there's definitely different types of diets, and many of you have maybe even tried these diets in your lifetime, such as the ketogenic diet or the low carb diets, or maybe you're doing these new intermittent fasting ones, which are very trendy right now. Or maybe you've just tried fasting, whether for religious purposes or even for just your own health purposes. And so we know that there's all these different types of diets, and the literature right now is emerging that some of them are actually pretty good. Diets can increase the efficacy of a number of treatments, such as chemotherapies, targeted therapies. So targeted therapies are things that sort of go after a very specific enzyme within the tumor. So like a HER2 antibody, or PI3 kinase inhibitor, or something like that. And then also, obviously, the new one is immunotherapy. Which is really interesting, because the bacteria in our guts, we know, are really important for keeping a healthy immune system. And the communication between those bacteria that live inside our colon and our normal immune cells, like T cells and B cells that can fight a tumor, are constantly communicating. So the bacteria are communicating to the T cells and back and forth. Because if you think about it, that's a bacteria that's living in your body. And why isn't your body trying to attack it and kill it? But a lot of that has to do with the barrier that exists in your colon, so the T cells can't actually go after them, and it keeps them separate, but they still release things and communicate to each other. And so it's really interesting that a number of those diets that sort of regulate our microbiota that live in our colons can also affect our immunotherapy responses in our patients. So that's something that's really being explored right now. But I voted this one up here because I think the best data that I'm seeing emerging has to do with fasting and intermittent fasting. And I think this probably is where I've seen the most convincing data, is what I'll say. And also in specialized diets. And some of the data with the ketogenic ones and low carb ones are a little bit more, so there's been some that show that it does work, and other ones say it doesn't work, and some that say it actually makes it worse. So, and also with caloric restriction, we find that this one's going to be really difficult for patients to do, because you're constantly under stress, and you're constantly eating less calories than you should every day. And so this one's not really optimal for patients. And a lot of the patients were actually getting quite sick in the trials with caloric restriction. So I think one of the ones that's emerging as really interesting is fasting and intermittent fasting. And the way this works, and what I think a lot of people are saying, is that you can't fast all the time. Obviously, that's not a possibility. But what they're really realizing now is that if you fast just prior to and during your chemotherapy, that you can really increase the tolerability of the treatment. And so a lot of the toxic side effects seem to be reduced. And also the efficacy of the treatments themselves seems to increase. And there's a number of reasons for this, but it's also a really fascinating area. And what they're suggesting now is that maybe it might be a good idea to sort of go through some type of intermittent fasting just during the treatment, and then stop the fasting, go back to your normal diet for a few weeks. And then if you go back to the clinic for more treatment a few weeks later, again, start the fasting just a day or two before, and then do it throughout the treatment. And then once that's done, again, go back to your normal diet. And this sort of cycling can actually, so like I said, alleviate some of the tolerability of the treatment and some of the toxicity and side effects. So, so this is all very, very new data. And so I think none of this is sort of ironed out yet. And I think there's a lot more work that can be done in this area. And what was really great today to see is that NIH, so the National Institutes of Health, who fund a lot of the research in the United States, the NIH just said that they are going to have a 10-year initiative to better understand diet and disease. And so they're going to be putting a lot of funding into understanding how what we eat can impact our disease states. And can we use it as a therapy? Can we somehow combine fasting and intermittent fasting with our chemotherapy, or our radiotherapy, or immunotherapy? And can we get better outcomes for the patients? So I'm really looking forward to that, and really seeing a huge explosion in this area. I think a lot of the studies that we're seeing now are just starting to emerge. And so, like I said, please take these with a grain of salt, pun intended, and sort of understanding that, I think there will be some type of level where we can have specialized diets for each treatments based on the patients and their tumors, and it will have to be individualized. So that's what I always say. You should always consult your physician or oncologist. Don't do anything on your own. Definitely do it with some informed consent from everyone else as well. It can depend on your cancer that the person has, as well as the underlying genetics, as well as your overall health. So obviously people can be diabetic and they can have other type of metabolic diseases that are going on while they have cancer. So all this needs to be taken into account. And can diet be used to prevent cancer? This is a much harder question to answer, but my advice to anyone that I talk to is that you stay away from doing too much of any one thing. So you don't want too much sugar. You don't want too much high fructose corn syrup. You don't want too many fatty foods. You don't want too much alcohol. You really want to keep all of them at some type of moderate level in a balance. Just don't overindulge is what I say. And having a healthy balance of bacteria in your gut, as I said before, helps keep your immune system in balance. And so things like dietary fibers, foods that are rich in fiber, such as fruits, vegetables, as well as things like quinoa and beans and legumes. And of course, moderate exercise is always good, and at least some level of exercise. But using cancer to fight existing tumors or cancers, or using diet to fight existing cancers, I think is much more doable. But there still needs to be a lot more work done. But I do think that we'll be able to sort of incorporate these into the treatment regimens for patients in the future. In other words, can we reduce your high fructose corn syrup intake? Can we reduce your alcohol intake when you're on there? For instance, if they sequence your tumor with these new really advanced technologies where they look at all the genes that are in your tumors and they see that the tumor has a lot of acetate metabolism genes, then maybe, reducing fructose and alcohol in your diet would really help if you were to take like an inhibitor or something that would fight acetate metabolism. And I basically hope one day we can sequence everyone's genes and everyone's cancers so we can make much better informed decisions. I think one day we'll get there. But our goal as researchers is to translate our knowledge into well-informed, scientifically grounded, which is very important, right? To make sure that these are all grounded in science. And potentially create personalized dietary recommendations for cancer patients. I think this would be a really amazing thing that's inexpensive, could have a really large impact, and could be used across all cancers, because every cancer is going to be a little bit different, but why can't we change the diet for all these patients? So I think there's a lot to be done, but I think that's really hopeful, and I'm excited for it. So I'll just stop there and say thank you to everyone in my lab. So this is my lab here with all our Schug Lab shirts on. That's actually acetyl-CoA, which is made from acetate. So that's our favorite little thing that we study, and we're really all about acetate metabolism. And we also have students rotating in the lab. Thank you to advocates that I've worked with. So Meryl is the one on my Susan G. Komen project, but I've also worked with Jamie Lascala on applications to the Department of the Defense as well. And she's been great and really helpful. And then past members here, Joe Salvino, he's the one helping us make the acetate inhibitors. So we've been working closely with him. He's a medicinal biochemist, so he's really smart in designing those molecules so that they work really well. And then of course we study the metabolism using really cool instrumentation that's run by these people here at the Wistar Institute, Sinyal, Nicole, and Tom. So they let us study all the little metabolites, and they measure the acetate in the tumors for us and stuff like that. And then I thank all of my sort of funders, including Susan G. Komen with my Career Catalyst, but I'm also funded from the V Foundation, the W.W. Smith, as well as NCI, and I'm a winner of the NIH Director's New Innovator Award. So thank you, everyone, and of course, I'll be happy to take any questions.
Super.